Good afternoon, everyone. Thanks for toughing it out. We're almost there. My name is Kirill. I'm a postdoctoral fellow with Caddy in Dr. Kang's lab. And today I'd like to talk to you about pathologic neurotransmission in the SL mouse model. So I think we've talked a lot about this model today. And about the GABAergic synapse. In our lab, we've studied this model for a while. It has absence seizures, spike with discharges. It has certain behavioral deficits that we're finally publishing this year. And then has a prominent trafficking pathology that results in loss of GAT1 expression and GABA uptake. And so a big question for me was how all of this affects neurotransmission, inhibitory neurotransmission in particular. So first I focused on synaptic deficits. So I looked at literature, looking at knockout mice, and kind of how Michael was saying, in the knockout, the miniature iPSCs and spontaneous iPSCs, spontaneous events, smaller events, aren't changed. But several papers showed that profound change in evoked events, either a large evoke event, or in this particular paper, there was a difference in evoked event decay, and then if you have a compound one, we have multiple evoked events stacked on one another, then the change in decay in that knockout is even bigger. So as you can see compared here versus the compound. So I wanted to see if I could see something like this in the SL mouse model, and I looked in the heterozygotes. I looked on cortex because that's relevant to absence seizures. And I did see a difference in decay, wild type in blue, heterozygote in red here, so the events are slower. And also in the homozygote saw a difference in decay. And then when I washed on GAT1 blocker tiagabine, I saw that in the wild type I could also affect decay, so it would get prolonged. In the heterozygotes I could do that as well, but not to the same extent. So if you kind of overlay it, you can see where here's the wild type, here's either the wild type or the heterozygote with GAT1 block, and then without the block, heterozygote is kind of in the middle. I also wanted to see if this would be exacerbated by in a compound event where you have multiple evoked events stacked. So I did this, and this is one of the more recent experiments I've done. So with a single event, this is new data from the same cells. When I'd look at a single event, I would again see this difference between wild type and heterozygote, and then I would also see this difference if I had it stacked. I broke this data up by bins to see how, depending on the amplitude of the event from here to here, if the decay differences were different. And I also looked separately for evoked events and compound evoked events. So essentially what I found is wherever I looked, there was either a trend or significant increase in decay, but I wouldn't necessarily say that these compound events were much longer than the evoked ones. And I think one of the reasons for that could be like when I was doing a similar experiment looking at successive stimuli in the wild type versus heterozygote, I could see a difference in decay, but still it was close to returning to baseline, whereas if I blocked GAT1 completely here with those blockers, then that's when summation really started occurring a lot more. So I think that's what's happening there. And I also looked at spontaneous events, and I saw actually in this mouse model in my data, I saw a change here in decay, both in the heterozygote and in the homozygote. You see right here. And of course, I wanted to see if PBA could restore this phenotype that I found. I did 2 different treatments, 7 days and 28 days. For these large evoked events, I actually did not see a rescue. So the light green and dark green are both heterozygote that's been treated. Light green is 7 days, dark green is 28. But as Caddy mentioned earlier, for spontaneous events, the longer treatment did seem to start returning these events closer to that sharper decay that you see in the wild type. I also tried acute application, given that PBA may have allosteric effects. But in my experiments, I did not see an effect either for evoked events, for wild type, or for SL heterozygotes, or for spontaneous events when I washed on PBA. I did 10 millimolar here. So that led me to conclude that PBA treatment partially restores this phenotype, and then this prolongation of synaptic responses, that's a quite stable phenotype that I'm observing in these mice, particularly for the larger evoked responses where uptake seems to matter for clearing that GABA, as opposed for smaller, faster synaptic spontaneous events where diffusion might play a big role as well. So then I wanted to look at some other things. I wanted to look at tonic inhibition, which in the knockouts, in the GAT1 knockout, it's been found in a whole host of regions. And as Caddy mentioned earlier, we do see a change in uptake when we do synaptosomal, gliosomal uptake assays, where uptake we know is reduced in these mice. We've also done microdialysis studies where we see that GABA starts to accumulate, though this occurs later with age, but it starts to accumulate in the brains of these mice. So I wanted to see if tonic currents are elevated, and in the regions where I checked, I actually, and this is several years old, I've shown this data to you guys before, I didn't actually see a change. In cortex, I looked at 2-3 months, I looked at 7 months, I looked at homozygote versus wild type, homozygote heterozygote wild type, I didn't see a change. And I looked in the thalamus as well, and thalamus is known to have a pretty large tonic current, which I did observe, but it just wasn't different in the heterozygote versus the wild type. So I wasn't quite sure, like I had a good feeling that tonic currents are involved in this disorder, but I wasn't seeing them. Then I came across this paper that gave me an idea from the experiment. In this paper, Pandit et al., done by this Korean group, essentially what they do, they look at granule cells in hippocampus, and they look both in development and in the adult, or at least young adult. And so what they're doing here is at first they're washing on a low concentration of GABA, so they're seeing if there's sensitivity to GABA elevation. And then they're blocking GAT1, and then they're seeing if there's sensitivity to GAT1 block. And then, of course, they're just blocking GABA-A receptors, and then just seeing what's the overall tonic current. And this is really good for looking at different aspects of this model, right? We can look at sensitivity to what happens when you increase the GABA amount. Then we look at sensitivity to what happens when you block GAT1, as opposed to simply looking, is there tonic current, is there not a tonic current? And another important thing here is they also looked at a developmental time point, and in this particular region, at this time, this region is actually sensitive. Like if you wash on a bit of GABA, you do get a tonic current from it. And this region is also, in this region, GABAergic currents are particularly important tonic currents for development as well. I wanted to start doing this experiment. I'm kind of in the middle of it. I haven't done any developmental stuff, but I've started doing the adult experiments, and it's pretty interesting what I'm seeing so far. So you can see here, this is in the wild type in blue. So I'm washing on GABA here. I don't really see much of a change. You might see a bit, but there's not too much. When I wash on tiagabine, then there is a dip here in the baseline, very clear, which then goes away, and it mostly returns to this baseline when I block GABA-A receptors. And that's not really what I've been seeing, at least in some of the animals that I've looked at, some of the cells in the SL. So I'll wash on GABA, and I expected that these cells would be more sensitive to it. I thought maybe I'd see a tonic current here. No, I did not. So they were, and maybe if I'd wash on more I would, but here I didn't. But then I washed on tiagabine, and here I expected these cells to be perhaps less sensitive to it because they're already missing a lot of GAT1, and the concentration that I'm using this at should be, it's like 3 orders of magnitudes higher than EC50. So it should really block all of GAT1. But I would see a very prominent current, as you can see here with tiagabine. One cell, here's another cell as well. And then when I would block it, then the baseline would go up and wouldn't just cancel out this current, but it would actually go above my previous baseline, as you can see. So it would reveal that there was a tonic current here at rest greater than in the wild type so far. So this is the summary of those results so far. So you can see these numbers here, they're the medians. So if we go left to right, at first this is so the blue is the wild type, the red is the heterozygote. So at first I wash on GABA, and the tonic current change here is quite small, so 3, 4. But then I start washing on tiagabine, and this is the first minute here, and it's the second and third, so it takes it a little bit to take effect. But you can see at the second and the third minute here, I get about 20 picoamp current in the wild type, and it's more like 35 in the heterozygote. So it's present in both, but it's higher in the heterozygote. And then when I wash on bicuculin, mainly in the wild type, it just takes away this current that came on with tiagabine, but in the heterozygote, it overshoots it and reveals that there was also presence of a tonic current before I washed on anything, practically. So this is pretty interesting. I'm looking forward to continuing this. My ends are they're kind of halfway there, so I have looked at a number of cells, but I want to see if this trend sticks, because it's not entirely what I expected to find, but this elevated tonic current, it does make sense. And then another aspect of GABAergic signaling I was looking at is whether GABA is depleted presynaptically. So as I've already shown you, these traces I've been looking to see at paired pulse ratios to see if the subsequent responses become significantly smaller in amplitude as you deplete presynaptic GABA. I have not seen that, which is quantified here when you look at amplitude ratios. What I've seen is just consistent differences in decay, but I want to come back to this with more intervals between those events, because that's the best way to do this type of experiment. And I started doing GABA depletion experiments where I am basically looking at the evoked response I get a good baseline, and then I deliver high frequency stimulation to release all of the presynaptic GABA, and then I see how long it takes for that to replenish, and then if uptake is inhibited and it's important for replenishing, it should take longer. And that seemingly is what I'm seeing so far. So the black here is the wild type, and the red here is the heterozygote, and it looks like it takes longer to recover to this baseline right here that we started at. But this is still an experiment that I want to refine, because I had a lot of scatter. Like if you actually look at the data points, so some cells never recover, some kind of over-recover, and I based this off of a cerebellar study, which was a lot cleaner. So I think half it being cortex, half maybe needing to optimize my stimulation parameters, I might be able to get tighter recoveries that could help me analyze this better. Okay, so I want to come back to this model that I'm showing you here. And today we talked about changes in synaptic inhibition, in tonic inhibition, and then presynaptic changes. So I think synaptically, I've done a lot of experiments that show quite consistently that decay is prolonged here. And right now, I'm mainly looking at tonic currents and depletion of GABA, and I've shown you the most recent data I have for that. So thank you very much. I'd like to thank my lab. Here we are, some pictures of us. We've been having potluck lunches, some in the front, because they make me take all the selfies for them. And we have a lot of undergraduate students. I'm only showing here 3 that have been helping me, and we have a lot more. So thank you to all of them.